morning, students. Today we are going to learn about cellular metabolism. First topic, carbohydrates. We all know that carbohydrates are the sugars which are naturally occur compounds called carbonyl compounds, that is aldehydes or ketones, which contains several hydroxyl groups. It also includes the derivatives which produce such an compounds on and when it undergoes the hydrolysis. They are the most abundant organic molecules in nature and also referred as saccharides. The carbohydrates which are soluble in water and sweet in taste is called as sugars. Next, carbohydrates are divided into four types, that is the monosaccharides, disaccharides, oligosaccharides and the polysaccharides. It is divided based on the sugar components. If the in monosaccharides, if there is only one sugar, it is divided into monosaccharides. Examples are glucose, fructose and galactose. In this disaccharides, if it contains two sugar compounds, it is known as disaccharides, and the examples are sucrose, lactose and maltose. Next, oligosaccharides, it undergoes 3 to 10 sugars, which is examples of raffinose and stachyose. The polysaccharides which contains about 10 components of sugars is called as polysaccharides. The examples are starch, glycogen and cellulose. This is the structure of the carbohydrate. This is the monosaccharide, this is the disaccharide, two sugar molecules. This is the polysaccharide, more than 10 is called as the polysaccharide. Monosaccharides. These are the simplest group of carbohydrates, also called as the simple sugars, and since they cannot be further hydrolyzed. These are colorless and crystalline solids that are soluble in water and insoluble in non-polar solvent. These are compound that possesses a free aldehyde or a ketone group. The general formula of carbohydrate is CnH2O. They are classified according to the number of carbon atoms they contain, also on the basis of functional group present. The monosaccharides, thus with it based on 3, 4, 5, 6, 7. Likewise, it is divided into trioses, tetroses, pentoses, hectoses. Likewise, oligosaccharides. Oligosaccharides which yield 2 to 10 molecules of sugar, same or different monosaccharides on hydrolysis. These oligosaccharides are the monosaccharide units which are joined by the glycosidic linkage. Next, based on this number of monosaccharide units, it is further classified into disaccharide, trisaccharide, tetrasaccharide. Likewise, in disaccharides we have 2 sugar molecules, in tetrasaccharide 3 molecules, in trisaccharides it has 3 molecules, and in tetrasaccharides it has 4 molecules. The general formula of this disaccharide is CnH2O and -2. These examples are sucrose, lactose, maltose, etc. These are polysaccharides, also called as glycans, it contains more than 10 monosaccharide units. It yields more than 10 molecules of monosaccharides on hydrolysis. Each polysaccharide differ from each other based on the identity of their recurring monosaccharide units, that is like in the length of their chain, in the type of the bonding they have, like in the linking units, and the degree of their branching. They are primarily concerned with the two important functions, that is structural functions and the storage of energy. It is further classified into the two types based on the hydrolysis. First one is homopolysaccharide, next one is heteropolysaccharides. Homopolysaccharides, when it undergo hydrolysis, it gives the same type of monosaccharide units. If in the heteropolysaccharides, on undergoing hydrolysis, it gives the different types of monosaccharides when undergoing hydrolysis. Homopolysaccharides examples are starch, glycogen, cellulose and pectin, and the heteropolysaccharides are hyaluronic acid and chondroitin. Next, the structure of glucose. Glucose is named as the D plus glucose, and which means the D represents the configuration, whereas the plus represents the dextrorotatory nature of the molecule. It has the ring structure and the open chain structure. The ring structure of glucose can explain many properties of glucose which cannot be figured by the open chain structure. The two cyclic structures differ in the configuration of the hydroxyl group at C1, and anomeric carbons such isomers, that is alpha and beta form, are known as anomers. The cyclic structure is also called pyranose structure due to its analogy of pyran. This is the structure of glucose. This is the open chain structure and this is the cyclic structure. Next, classification of protein and general characters of amino acids. Proteins as the most biological macromolecule which is occurring in all cells. This is the versatile organic molecule which is present in all living system, in all thousands of different kinds, ranging in size and relatively small peptides to the large polymers. Proteins are the polymers of amino acid covalently linked by the peptide bonds. The building blocks of proteins are the 20 naturally occurring amino acids. Thus, proteins are the polymers of amino acids. Due to the different rearrangement of amino acid, the structure of protein is divided into primary, secondary, tertiary, and quaternary. If the covalent linkages of one, this is the primary structure of the protein. This is the secondary structure of the protein, in which the linear peptide chains fold either in a alpha helical structure like coiled or pleated structures like sheets, which contains the hydrogen bonds. And next, tertiary. In this tertiary, the arrangement of and interconnection of proteins is have specific loops and bends, and also contain hydrogen, ionic and disulfide bonds. In quaternary structure, this structure in proteins contain more than one peptide chain. Next, classification of proteins based on shape, constitution, and nature of molecules. On the structure, on the classification of shape, it is divided into fibrous and globular. In fibrous, they are the proteolytic enzymes which are insoluble in water, and also they are coiled and exist in thread-like structures called to form a fibers. Examples: collagen, actin, myosin, keratin in hair, claws, feathers, etc. The globular protein, these are unlike fibrous proteins which are soluble in water, and they are made up of polypeptides that are coiled about themselves to form oval or spherical molecules. Example: albumin, insulin, and hormones like oxytocin. And next, on the basis of constitution, proteins are divided into simple, conjugated. These proteins are made up of amino acids only, albumins, globulins, and prolamins. And then based on the conjugated proteins, these complex proteins are combined with the characteristics of non-amino acid substance, such as called as prosthetic group. These are the following types. These are of following types: nucleoproteins, which is the combination of protein and the nucleic acid; mucoprotein, protein and carbohydrates; glycoproteins, proteins and carbohydrates which are less than the 4%. If they are greater than the 4%, they are called as the mucoproteins. Chromoproteins is the combination of protein with colored pigments. Chromo means color, and lipo means protein with lipids, and metalloproteins with metal ions, and the phospho combination of proteins with phosphate group. What are the general properties of amino acids? They have a very melting and boiling point. These amino acids are white crystalline solid substances. In taste, few amino acids are sweet, tasteless, and bitter. Most of the amino acids are soluble in water and are insoluble in organic solvents. Next topic: lipids. Classification of lipids. Lipids are classified into two main classes: non-saponifiable and saponifiable. In a non-saponifiable, lipid cannot be disintegrated into smaller molecules through hydrolysis. It includes the cholesterol, prostaglandins, etc. In saponifiable, they comprises of one or more ester groups, enabling it to undergo hydrolysis in the presence of only base, acid, or enzymes includes like waxes, triglycerides, sphingolipids, and phospholipids. It is also divided into simple lipids. Simple lipids like fats, waxes. In fats, these are the esters of fatty acids with glycerol. Oils and fats are in the liquid state, and waxes, which are the esters of fatty acids with higher molecular weight of monohydric alcohols. Next, complex lipids. Phospholipids. These are the lipids containing, in addition to fatty acids and alcohol, phosphate group. They frequently have nitrogen containing bases and other substituents. Example in glycerophospholipids, the alcohol in glycerol, and in sphygmophospholipids, the alcohol in sphygmosine. And next, other complex lipids called sulfolipids, which are called as aminolipids, lipoproteins. Next, derived lipids. They include the fatty acids, glycerol, steroids, and other alcohols, and fatty aldehydes and ketone bodies. Hydrocarbons, lipid soluble vitamins and hormones, because they are uncharged. Acylglycerols, cholesterol, and cholesterol esters are termed as neutral lipids. These compounds are produced by the hydrolysis of simple and complex lipids. Next, enzymes. Classification and mechanism of enzymes. What is meant by enzymes? Enzymes can be defined as it's a biological polymers that catalyze biochemical reaction. These are some of the types of the enzymes, which are oxidoreductases, transferases, hydrolases, lyases, isomerases, and ligases. Only the names of the types denotes their functions. Like the enzyme oxidoreductase catalyze the reaction, oxidase reaction, where the electrons tend to travel from one form of a molecule to the other. Transferases. It helps in the transportation of functional group among the acceptors and donors. And next, hydrolases. Hydrolases, which is in the word like that, it is a hydrolytic enzyme which catalyze the hydrolysis reaction by adding water to cleave the bond and hydrolyze it. Next, lyases adds water, carbon dioxide or ammonia across double bonds or eliminate these to create double bonds. Isomerases. This enzyme catalyzes the structural shifts present in a molecule, thus causing the change in the shape of a molecule. Next, finally, the ligases. It is the enzyme that are known to charge the catalysis of ligatic process. Next, this is the mechanism of enzyme action. This is the substrate and this is the active site. The substrate entering the active. This is the picture of substrate entering into the active site. This is the active sites of the enzyme. This is the enzyme. This is the active site. This is the substrate. This is the image of the substrate entering into the active site of the enzyme. Next, the enzyme substrate complex. This substrate is attached to the enzyme when it is getting attached, the enzyme changes its shape slightly as a substrate binds, and this complex is called as the enzyme substrate complex. And next, this is the enzyme substrate complex. Again, it is the product is leaving the active site of the enzyme. Thank you.